Our life science grade 12 2025. So I'm a little bit happy that we started together, we have concluded together, thank God. I'm also happy that you are here to check and see how far we have gone. I thank everybody who tried to join our classes. Before we start, let's do this and we see what. I know that you have been hearing that sound. It shows that the sign that the ThunderEDUC has arrived. It means that we are moving like a bullet. So if you want your marks to increase like a bullet, you have to join ThunderEDUC. You can tell your brothers, sisters, and others. Anyway, let's start without wasting time. I hope everyone is hearing me. Before I start the paper, I'm going to use 1 or 2 minutes just to remind you something very small of what we did last time. All right. Let's start. Remember our prediction or remember our things we did? When I checked the paper, I've not even I've not taken all the time to check the paper versus to what I told you to read about our scope 100% project. We see that this was there, monomers of DNA, nuclear, other types of nuclear DNA, other debates of profiling this structure. It was related to that. They brought about this ribosome. They ask you even it was the same circle. They brought almost everything. When I check, they brought this, which is like this. When I was discussing, I tried to explain. They brought Down syndrome. I asked you to show you how to do that. They brought about the definition of this. They brought this, but in a different angle. But I explained it. They brought all this. This one they brought exactly the same question about the same disease. They brought. Actually, when I saw the paper, I saw that people who used our discussion we did, I think you got it right. And we pray that you get all this. They brought about it. They brought about the locus. They brought about the species. They brought... what should I say? Why shouldn't I say? They brought this time. They increased the generation to 60. Then I used the one for 30th generation. They brought these one skulls. They talked about the comparing the skull of the specifically the skull, not any other part. They brought about the 3 factors about bipedalism, what causes. Yes, they brought it. What can I say? They brought these calculations. They brought about these calculations exactly. They brought about the out of Africa hypothesis, other factors which is about the fossil to use it. Actually, you guys, you did. I hope you did a great job. Let's see how we're gonna go through the question paper, and then we see how much you have got. We thank everybody who tried to join our class. And we tell you that this is not the beginning and this is not the end. And remember, not the best in class is the best in life. Not the person who gets the most marks is the one who succeeds in life. So please don't be worried about the mark. The mark do not determine the life. However, the mark can facilitate the life. So let's start, guys, without wasting time. All right, we are starting. The question is saying that, I don't read the instructions. They're saying that gene. What's a gene? A gene is a section of DNA that controls a particular characteristic. That is it. Ne? So it means that set of chromosome, nope. They're saying a set of chromosome segment. That is it. Segment, but is it? Coding for a particular characteristic look to be much more correct. They're saying the haploid number, nope. They're saying triplet bases that code for an amino acid. It's not just a triplet. Yes, there are triplets, but not just one triplet. So nope, it's supposed to be a segment. A segment is made up of many triplets. So B becomes the most correct answer. We go to the next question. Let me check something here. Segment. Cool. They're saying that significance of DNA replication, we talked about it. Yes, we talked about it. In the cell formed at the end of mitosis. The significance of DNA that is formed at the end of mitosis. Remember, mitosis, the cells are the same. So genetically identical chromosomes or cells. So that looks to be the most correct answer. Half the number, nope. Genetically different, nope. Half the double the chromosome number, nope. These other things are the importance of meiosis. The remaining one is the importance of mitosis. So A looks to be the most correct answer. Why am I shaking? Don't shake, Buffett. Which one of the following statement is a feature of punctuated curriculum? I told you, define about the punctuated curriculum. Okay, remember we said that long period of no or little change with alternating with rapid period of short period of rapid change. So we have to look for that. Individuals organisms adapt to changes of environment. Nope. The inheritance of acquired characteristics. Nope. They're saying that the long period of little or no change. That's what I told you in that. Some of them I didn't even tick them because our spot was around 98. We shifted from 95. Yes, for the case of paper 1 was 99%. At least last year we were 95%. This time it was 98 paper 2 and 99 paper 1. Yes, so tell your brothers and sisters that the thunder moves like a bullet. All right, we are continuing to the next question. So this one looks to be the most correct answer, which is C. Long periods of little change and then or no change at all, but the periods are supposed to be longer. Remember that we say that it's like a ladder whereby you have long period of no change and short period of rapid change. Long period of no change or little change with rapid period of with the short period of rapid change. So that is punctuated equilibrium. We talked about hemophilia. That was also there. That the disease characterized by lack the disease which is characterized by the disease which or disorder which is as a result of failure of the blood to clot is regarded as what? And then we also brought it as a question that hemophilia is a sex-linked disorder. Don't want to talk about it, but I just want to show you something here. They said hemophilia is a sex-linked disorder, and then here they said a mutation caused by a mutation on X chromosome, meaning that it's a sex-linked disorder. Yes, sex-linked disorder. So B looks to be characterized by inability or distinguish between red and nope. That is color blindness caused by non-disjunction. That nope. That is the Down syndrome caused by allele carried on orosome, not orosomes, sex. So meaning that B looks to be the most correct answer. Orosomes are body cells. We go to 1.5. Which one of the following is an example of heterozygous? Hetero means different. Different may be X and X. X, okay, D and small d, something like that. Okay, they're saying that if you look at hetero means different, so it look at this, it's the same. So this is homozygous, this is also homozygous, this is sex linked, this is non-sex linked, so it's out. This is homozygous recessive, this is out. So C looks to be the most correct answer. RW, this is heterozygous, so that is to be the most correct answer. We go to the next question. We brought this question, but in a different version. Did you pass it, guys? Let's go and then you find out what is it all about. Which one of the following is the combination? Which one of the following is a combination correctly? Okay, here we say that this diagram below shows what? DNA replication. So okay, now I know that this DNA replication. So they're asking you that which one of the following combination correctly identifies molecule 1, 2, and 5? 1, 2, 1, 4, and 5. Okay, let's go to molecule 1. What is molecule 1? That molecule 1 is this molecule. That's the reason why I'm giving you molecule W. Done. All right. Life is good, man. Don't worry. ThunderEDUC, M.SAIDI. Can't cook and then the food doesn't become nice. The food must become nice. When I cook. When I cook for you guys, my students. All right, we are saying that's it. So it's a nucleotide. So I know that it's a nucleotide. So whenever I see a nucleotide, I mark nucleotide. So this one, so B is out. So if you wrote B, you are gone already. Number 4. Where is number 4? Number 4 is, since they say it's DNA replication. See, they say DNA replication. So what is number 4? Number 4 is deoxyribose sugar. Deoxyribose sugar is the same question here. I asked you that what is Y. All right, we continue. Then they're asking you the next question, the number 5. Number 5 obviously is the phosphate group, and is the same question here, which is XC. They just changed the numbers, and then so A looks to be the most correct answer. We go here. Which one of the following combination is correct for number 2 and number 3? Number 2 and number 3. So let's go to number 2. Where is number 2? You see here, they brought it. Okay, number 2. I gave you an idea of using this. By using this, you guys, you remember that what is this thymine? What is this? It said it's adenine, but you have to look at, because here is thymine, so you look at it and then you are able to identify what is V. So basically that's what they're asking you here. So what is this? So you need to understand the concept that, this. If this is 2, what about, okay, now what about this? So if I can know this, remember this thing we are together. So this is the first nucleotide, this is the first nucleotide. So if this is A, automatically this is A. The other strand must be T. And if this is T, automatically number 2 is A. So I'll come back here and I say that number 2 is A. So I know that, number 2 must be A. Okay, then I go to the second one, which is number 3. Number 3 is the same story, is the same idea which I gave you to use. Hey, I told you. We are saying that, hey, Akra, I need to show you how did we get to the answer. Hey, don't rush, buffet. Then we say that, we continue and then we say that number 3. Where is number 3? Okay, number 3 molecule. Number 3, they say that number 3 is 1, 2, 3. So I count 1, 2, 3. So if this is thymine, automatically this is gonna be adenine. Therefore, you're gonna be adenine. That's the answer. So B becomes the most correct answer. All right, we go to the next one. Question 1.8 to 1.9 refer to the information in the dihybrid cross. Okay, dihybrid cross. Did I finish the objectives? Not yet. Let's talk about the dihybrid cross. Actually, even this one was almost similar to the question which I gave you in the previous. All right, let's continue. So you guys, sometimes I have to a little bit to explain because there are some other people gonna be watching this video. For example, those are upgrading, those people who are going to be using it for next year. That's why I don't just need to rush. However much I'm rushing, but I don't need to rush. I need to show you where is the answer coming from. Yeah, it's not just for guessing. All right, in certain plants species, on one gene controls stem color and another controls height. This is the height, and then the other one controls the height. Okay, they're saying the stem can be brown. Brown is capital B and red for small b. While the plant with the height, tall is capital T, short is a small t. All right. When plant 1 was crossed with plant 2, plant 1 is brown. So we see that brown is small b, and then no. Brown is dominant over what? Brown is capital B. It's gonna be capital B. It can be capital B. All right, let's look at it like that. Or capital B small b, capital B small b. Did they say it's homozygous? Okay, we shall see. And then they're saying that red means that this is small b, and then they're saying that this is tall, it can be capital B, capital T, or capital T small t, and then short automatically small t. All right, let's continue and find out which one of the following represents the genotype of parent of plant 1. Plant 1 is here. Okay, it must be having small t. I need to look for small t. So small t. So these one are out. Oh, all right. And then plant 2, I can look at red is small b. So I have to look for small b. So this one, remember this one I said is correct, but it's not small b. This one I said is correct, small b. So this one looks to be correct. So this one is already, yes, is correct, but is already out from this side. So this one is also out. So the most correct answer looks to be B. So B becomes the most correct answer. All right, they're saying that the number of possible different genotype expected in the offspring in cross this. Okay, when you cross them, what do we expect it to be? When you cross them, what are we expecting it to be? So it's gonna be let me scoop out the genotypes. Is B, small t, and then this one is going to be small t, small b. I'm reading it from here. Yeah, I'm reading it from there. So it's small b, capital T, small t. So since this one are the same, so I'm going to take one. And then this one is going to be, okay, so it's going to either be, capital B, small t, yes, or is gonna be small b, small t when I combine. Okay, some of you didn't get how did it come about. I'm saying this is how it was. This goes with this, is gonna be capital B, small t. This goes with that, is gonna be capital B, small t. This is gonna go with that, is gonna be small b, small t. This one is gonna go with that, is gonna be small b, small t. So automatically I'm gonna say capital B. You see, they said different because the key point is saying different. So this one and this one is the same. So I'm gonna take capital B, small t is different, and then this one also is gonna be small b, small t. You understand? And then now this one is gonna be It's the same story. This is gonna be small b, capital T. This with this is gonna be small b, small t. This one is gonna be, you see, small b, capital T. This one is that, small b, small t. So you see that this and this, they are the same, so I cancel out one. This and this are the same, so I cancel out one. So automatically my genotypes are going to be capital B, yes, small t, yes, this one, and then small b, small t. You understand? And then now let me clear up this one. Then this one is gonna be small b, capital T, and then small b, small t, like that. So basically when I cross, you'll find how many spaces do I have? 1, 2, 3, 4. So there are 4. So the answer is A. So if you cross them, you're gonna form 4 different individuals. You understand? When I cross, that's because they said from the cross. You understand? When you do, they're gonna be 4. So the correct answer becomes A. So that is it. Then we go the next question. Let me clear up here. They're saying that a rare form of rickets. Rare form of rickets is caused by a dominant allele on the X chromosomes. I explained this yesterday very well. Which one of the following statements is true about the inheritance of this form of rickets? Which one of the following is true? If the father is affected, all the sons never, because the father will, to the sons, he will give you the recessive, the Y chromosome. So this is out. If it will be expressed more frequently in males than females. Not dominant allele, that is only for recessive allele. Out. If the father is affected, all the daughters will be affected. Yes. Because it's caused by dominant allele. Look, guys, if this is capital D, and this is why, because the father is gonna be giving to the girls an X. So I don't care what is on these ones. Yes. So if this goes with this, it's gonna be X, capital D. Whatever is coming from this side, I don't care. Then this one to this is gonna be X, capital D. You see, and another X. Whatever is here, I don't care. So you see that the females. Because now if I cross this, it's gonna be a male. You see? So the father will not give any allele to the male. It's only the mother. So basically all the daughters will be affected with the disorder. Therefore, C looks to be the most correct answer. If it will be expressed in a female, since they have two, it will only be expressed in females, since they have two. No, even males, it will be there. Even males, they will have the dominant allele. Who got 20 out of 20? Okay, who get 18 out of 20? Who got 18 out of 20? Oh, I see some people getting the yes. Thank you so much, guys. Let's go to terminologies. Terminologies. Where are you? Who got all the marks in terminologies? Let's start. Bond formed between two amino acids. Peptide bond. Peptide bond. Remember, we don't call them polypeptide bond. Peptide bond. I told you yesterday. An organelle that contain DNA, which is used to trace the female ancestry. We said it's a mitochondrion DNA. Mitochondria is a mitochondria. There's an undifferentiated cell that have the potential to become any type of cell division. I don't want to go back to show you these things. I don't want to go back because it's going to be taking a lot of time, because you saw those people already. This question also was there. Even this one was there. Okay, let me just continue. We call them stem cells. not cells of the stem, stem cells. All right, a change in the sequence of nitrogenous bases resulting in the formation of different proteins. How do you call that? We talked about it. We call it a mutation. Mutation, but they're saying sequence of nitrogenous bases. So it's better to say gene mutation. Yeah, I hope they will mark gene mutation or they will mark mutation. Yes. All right. Natural shape of DNA. We have been singing a song on this. Double helix. Double helix. Helix. Okay. Permanent disappearance of species from Earth. Even a grade 10 can answer this one. Extinction. Extinction. The type of variation which intermediate. We talked about it and give an example of a head and skin. Yes, we call it continuous variation. Continuous variation. Variation. Yes. They're saying an allele that is expressed in the phenotype in heterozygous. We told you yesterday the same thing. Is expressed in the phenotype of heterozygous state. We call it a dominant allele. And then someone was saying, no, it's not allele, it's allele. And I say, okay, it's allele, whatever. Dominant allele because it expresses itself in the heterozygous state. All right. Position of a gene on a chromosome. Hi. Hey, how do you call that? We said you call it a locus. So what I did is I say, what is a locus? So that you know what is the position of a gene on a chromosome, position of an allele on a chromosome. We call it a locus. Locus. Good. I see people, they are bringing a lot of locus buffet. All right, I see people are hot. Yeah. It means that you are at on our discussion when you're busy talking about. Guys, don't mess up. Always watch ThunderEDUC with M.SAIDI. You will never regret. Who got 9 out of 9? 9 out of 9. All right, we continue. Guys, we are counting distinctions, and I told you have to be better than me. That's the idea behind this. You have to be better than me. All right, we continue. They're saying that the phase of meiosis during which non-disjunction occur, we said it. Non-disjunction, the phase of meiosis during which non-disjunction occur. It occurs in both anaphase 1 and anaphase 2. So that's why I said that the failure of the chromosome to separate during anaphase. They don't say during anaphase 1 or anaphase 2. Or they don't say during anaphase 1. Or they don't say during anaphase 2. No, they say during anaphase. Or during anaphase 1 and 2. So both A and B. Both A and B. Alright, we continue. Representation of number, shape, arrangement of all chromosomes in the nucleus. How do we call that? We talked about it. It's a karyotype. And someone is asking me, why bring the karyotype? I knew. Alright. Alright, don't ask me how did I know, unless I don't have brain. Alright, we continue. The allele of genes separate into different gametes. I even ask you, state Mendel's law of segregation. I didn't want to talk about Mendel's law of independent assortment. Me, I wanted the Mendel's law of segregation because I knew. So state it. Alright, the answer is B only. And even this one is B only. Alright, we continue. Hey guys, hey, life is good. I know that you are killing this paper. Who got 6 out of 6? Life is good. Who got 6 out of 6? Yes. Who got 6 out of 6? You see? I told you. He's here. State Mendel's law of segregation. The locus is here. The dominance is here. The dominant. Let's continue, man. Let's continue. Let's kill it. We continue. I brought this for you to label. I know this one you killed it. I told you that once it's not in a prophase, you can't see the nuclear membrane. So A is a cell membrane. B is a this is a spindle fibers. Yes, Bipedalism. Yes, Bipedalism. I had a reason, and even I told you answer that. I had a reason. Okay, we continue. What is C? Spindle, centrioles. Those are centuries. I had a reason. I told you to answer this. I had a reason. Not to. We continue. Which one of the following W, V, W, X, Y, and Z represent a cell during prophase? During prophase. We continue. Prophase. For that reason, have to drink some sip of water. All right, which one shows? I got you here. Prophase of mitosis. Now here, do you know what you're supposed to do? Someone said nuclear membrane. No. I told you this is not nuclear membrane, Buffett. I said this is the nuclear membrane. That's the nuclear membrane. This is the cell membrane. It's a cell membrane. This is the nuclear membrane. I explained very well that after prophase, that's why it's dotted, so that it's gonna disappear. After prophase, you don't have nuclear membrane. Where were you studying from yesterday? All right, we continue. Which diagram represents the cell during prophase of mitosis? Now here, let me clear it up. This is the part which made me lose the 99, the 100%. Yeah, they brought this question. I didn't think about it. Okay, this question, this one I'm going to answer. They're saying prophase of mitosis. In this case, remember in meiosis, now we need to identify. But I asked you to do the difference between mitosis and meiosis. So if you had that, then you are able to identify it. Okay, remember in meiosis, chromosomes align in homologous pairs. They are in homologous pairs. While in mitosis, the chromosomes are single. You understand? All right. Now let's check. They are pair. You see? This shows his prophase. Even this one shows his prophase. But one is prophase in meiosis and one is prophase in mitosis. Check this one is alone. 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 You see? So this is mitosis. This is meiosis. Because they have to align along themselves so that crossing over can take place. So, in this case, we're going to say W is mitosis. W. We call it as W. W is mitosis. Metaphase 1. Metaphase 1, they are arranged along the equator. Along the equator. This. All right. That question, this one was there. Even as you draw a cell after that. So it's gonna be Y. Prophase 1. Remember, mitosis doesn't have prophase 1 or anaphase 1. No. So this is prophase 1. So it's gonna be V. V here. W H Y V. How many chromosomes will there be in each cell at the end? Did I say this is a human cell? I told you, don't talk about human cells. How many cells will there be at the end of mitosis? The way they are, the way they will be. It's gonna be 1, 2, 3, 4, 5, 6. They're gonna be 6. 6 chromosomes. 6 chromosomes. So the answer here is 6. We continue the next one. Just a moment. Yeah. All right. We continue the next one. Just a moment. Yep. Okay, we continue next one. How many chromosomes would there be in each cell at the end of meiosis? Meiosis, remember, there are how many? It's 1, it's 2, it's 3, it's 4, it's 5, it's 6. At the end of meiosis, there is nuclear division. So it means that the number of chromosomes, they become half. So it's going to be 3. Hey, someone is asking me what is Z. They didn't ask you about Z. Did they ask you about Z? Why do you bother about Z? But Z looks to be prophase 2, because they already have the variations, you see, and the chromosomes are single, and because of this shows that is prophase. So that's prophase 2. Yeah, prophase 2. Yeah, and since it's 1, if there were 2, maybe it would have been hey, who got 8 out of 8? 8 out of 8. All right. Oh, who would have got 8 out of 8? All right, we continue to the next question. You guys, I hope you didn't disappoint me. Dun, 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 dun. Huh? Let's continue, guys. They're saying that the diagram below shows represents the stage of protein synthesis. Each amino acid and P2R is are coded by different amino codons on this messenger RNA. All right. The stage, identify the stage of protein synthesis shown above. You see it here, and then you ask, you say ribosome, and then you ask, you say ribosome, and then you ask. They're asking, they only brought this part. You see it there, and then you ask. So it is translation. 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 What is molecule 1? What is molecule 1? What is molecule 1? T. You see it's a T. Say T. So it's tRNA. It's tRNA. tRNA. What is molecule Z? We have been talking about it. Is a ribosome. Yes, I told you it. Yeah, when you were busy complaining about this, I said which molecule is 6. I had a reason, Bafeto. We continue. All right. Then they are saying that state the sequence of nitrogenous based on DNA molecule for codon. This the same things on that question. I don't want to go back there because it will be wasting our time more. State the sequence on number 3. Okay, number 3. Where is number 3? Number 3 is here. Number 3. They are saying that state the sequence of nitrogen bases on molecule for codon. Okay, number 3, DNA. So if this is messenger, so I'm just gonna change it. It's gonna be C, it's gonna be G, it's gonna be T, because it's DNA, so it's a T, not U. Done. We go to the next one, number 4. Number 4 is not there. Let me check. Okay, now you know what you're gonna do? We don't know what's number 4. We don't have any idea. They didn't label it here. We don't have any idea. So they want, what do they want? They want write down the sequence of DNA from number 4. So I need to find out what is number 4. So know what I'm going to do? I'm going to say CGU is a transfer RNA, anticodon. So CGU goes with what? C is going to go G, C, A. So what is CGA? So it's this one. So this one becomes 3. So that one is out. Then I'm going to go GCU. GCU going to go CGA. CGA. CGA is here. So this is number 5. Okay, let me go to this one now. GUA. GCUA is going to go with GAU. GA. It's going to go GAU. Where is it? GAU. This looks to be number 4. Okay, let me go to this one. AUG. A goes with U. U, A, C. Ah, yeah, this is number 2 because this is this one. So automatically this is number 4. Number 4 goes with this. But they want the one for DNA. So automatically it means that if this is CUA, it's going to be GAU. Then on DNA, it's going to be CTA. So this is what is going to be on DNA. So it's going to be CTA. So that is the answer. Someone is saying it's GU. It's going to be GTA. Why do you say GTA? No. Is it GTA? No. Okay, let me check it again. It's gonna be this one. Is gonna be C because this is anticodon. I told you it's anticodon. You change it to codon, and then you change it to code. That's what I told you yesterday. Then I say that if this is C, it's gonna go with the G. If this is U, it's gonna go with A. If this is A, it's gonna go with U. Wabona. Now I have to convert it into code, which is the DNA, because they want it on DNA. So it's C, G is gonna go with C, A is gonna go with T because it is DNA, and then U is gonna go with A. So it becomes CTA. All right. CTA is the most correct answer. We got 7 out of 7. Oh, which amino acid PQRS is coded by number 5? Which amino acid PQRS is coded by number 5? So we go back and find out what is number 5. Amino acid coded by number 5. Yeah? PQRS is coded by number 5. Oh, so we saw that this is 5 is here. So it's going to be P. Because this is CGA. So when I transfer it, it's going to be GCU, which is this. So it's P. So the answer becomes P. Hey, but guys, the paper was fair, ne? Yeah, the paper was fair. Someone saying 45 out of 50. Congratulations. Who got 50 out of 50? 50 out of, okay, 48 out of 50. Okay, 47. Okay, 46. I only see 45. Oh, I got someone for Lungelo Koza 46. Chayani say 46. 31, 44, 45. Thank you guys, ne? Thank you. Speciale. Is a specific. We're just laughing at. Oh, I have Ontile 48. Oh, Itum 50. Congratulations, guys, ne? Yo. I know that you guys were here at ThunderEDUC. Thank you so much, guys. Let's go to the second question and then we smash it. I told you, here at ThunderEDUC, we smash the paper. We don't play. Have you ever seen a thunder playing? No. We smash. Our slogan is smash it. All right. Alright, Sai is the best. Lolo is the best too, Priscilla. Alright, guys. The diagram below shows, I gave you this diagram, but it was in a different angle. I explained it, but it's in a different angle. That one. I had a reason for that. Alright, let's continue. Alright. Let's start. Identify the structure labeled A. Structure labeled A. Yes. Structure labeled A. That is a centromere. The central body. Centromere. Biological name for sex chromosome. We say they are called what? Gonosomes. They're saying that describe how the chromosome would differ in the somatic cell and of a male. Describe how these chromosomes will differ the gonosomes will differ from that of a somatic cell or of a somatic cell of a male. The somatic cell, then they will be identical, they will be homologous. homologous. You understand? Well, this one, it will be for gonosome, for this cell, it will be X and Y. It will be X and Y. So basically, the chromosomes in this individual describe how these chromosomes will differ. Yeah, there will be this one. There will be X and Y, which are not homologous. So the other ones, in the somatic cell, they will be homologous. So you are supposed to talk about X and Y. All right. An exchange of genetic material takes place at point P. Okay, we are listening. Describe the process. That's the process we talked about. Crossing over. Identify the phase during which this process takes place. Hey, guys, we say this what? Prophase 1. They're saying that. Give one reason why this process mentioned above is important. Guys, it's the same question, Z, I asked you yesterday when I gave you my scope. We kill two. We propose the scope for reality. All right, give one reason for it. Brings about genetic variation, and I told you it must be genetic variation, not just variation. Genetic variation. We continue the next point. Genetic variation. Explain how the structure of chromosome would differ at the end of meiosis. Explain how the structure of chromosomes will differ at the end of meiosis. Basically, each chromosome will consist of single chromatids which are non-identical. Yes. It means that, remember, they will be like this. Like this. And all of them, they will be non-identical. They will be single chromatids. They won't be two chromatids, no? Single chromatids, and these chromatids will be non-identical. Each chromosome will consist of single chromatids which are non-identical. This one I also give you the question. Non-disjunction of these chromosomes occur during meiosis and the gamete with an extra chromosome is formed. This is if this gamete is fertilized by normal gamete, by resulting zygote will not be normal. Okay, let's continue. Explain the genetic composition of the zygote. So basically, in this case, since there are 3 marks, they say non-disjunction of these chromosomes occurred during meiosis, and the gametes result in extra chromosome. So it means that I'm going to have a gamete which has an extra chromosome. Okay, cool. And it's formed this if the gamete is fertilized by normal gamete. So I'm going to have another gamete with the normal number. Okay. Result in the zygote will not be normal. Explain the genetic composition of the zygote. It means that the zygote, remember we say that the normal gamete, you need to tell us how many chromosomes will it be. It will have, the one which has extra chromosome will have 23 chromosomes. The normal gamete will have how many? Plus 1, which makes them to be 24. And then the normal gamete is going to have 23 chromosomes. When the two are fused, the resultant zygote will have 47 chromosomes instead of 46. So the Down syndrome, they're asking you, they just brought it in a short term, but I explained it, it was there in the questions I gave you, and I explained it, and I told you, take a screenshot. Take a screen what? Shit. All right. We continue the next question. All right. I hope, guys, you got all the marks there. 11 out of 11. We continue the next point. They are saying that, is there any question I skipped? Nope. They're saying that the diagram below shows. This one, I just used it from Distinction Material book, and then I told you what to do. The diagram below shows analysis of crime, okay, sample from the victim, sample, okay, from the suspect in their investigation. All right, in their investigation. In their investigation. Okay. They are saying that in their investigation, the police conducted the comparison of DNA to identify the criminal. All right. Identify the criminal. All right. Identify the technique which is being used. DNA profiling. DNA profiling. The technique. DNA profiling. Which suspect 1 or 2 was at the crime scene? So we see that this one matches with this, this 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 matches with this. Check, 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 man, check, man, check, man, check, man, check, man, check, man, check, man. So it's suspect 1. Suspect 1. Suspect 1. Give one observable reason for that. You you have to tell us that the DNA bands of suspect 1 matches with the DNA bands of that found on the crime scene. You understand? So that one of the bands matches with that of the crime scene. That's what it means. Therefore, it means that suspect 1 is the what? Is the problem, is the one who caused the problem. They're saying that give the possible reason why this suspect identified and 2 above might not be convicted if this evidence is used. Hey, don't forget that DNA is unique, but is only unique if a large sample is used. If a large sample is what? Is used. DNA is only used, you see, here is the question. You see it? There is the question I told you. Yes, suspect 1, suspect 2, suspect 3. And the answer was the middle one. This one and this one. You see, suspect 1. And I asked you the question Z, which followed there. So they just brought it. Alrighty. Okay, we continue. And then Alright. We're saying that basically DNA is not unique if a small sample is used. DNA is unique, yes. But this person might not be convicted because if a small sample is used, it might not be unique. That's why some parts of you can be resembling your father, you can be resembling your grandfather, you can be resembling your neighbor, you can be resembling your grandfather. You see, it doesn't necessarily mean that, your neighbor is your... I didn't say that. Yes, okay, if a small sample is used, so the DNA might not be unique. Then number 2, we call them the debates or the disadvantages. Then number 2, the person might be framed. What if someone collected his blood or somewhere and then went and then sprinkled it on the crime scene? It can be framed. Yes. So you can look at any other. Wabon. Human error. Someone is telling me even that one. Human error. Maybe someone who was drunk over the weekend has babalaza, and then in the morning on Monday, and then is doing the investigation, still even having babalaza. You think this person is gonna give you correct results? Okay. So it means that human errors can happen. Even I might not analyze properly. Oh, there is some contamination. There is a lot of things. Here is not like this the answer. There are a lot of answers you can talk about. Then they're saying that give 2 possible reasons why the suspect identified above. Okay, we're done with that. Explain how the technique. Yes, explain how do I use this technique to identify, to bring about the determination of this individual. How am I going to use this technique to identify the individuals? So basically, the DNA, we know that DNA evidence might be explain how the technique is used in identifying the paternity test. So basically here, what you need to do is to first identify the the sample of the child. You understand? Then you identify the sample of the individual. Then you have to compare it with the what? With the individual. If you're talking about the parents, if it's a father, all the bars which correspond with the mother, the remaining bars must correspond with the father, and then that will confirm that that is a father. Let me show you here. Yes, it's just here. Okay, it's just here. Paternity test. Yes, paternity test. Paternity test. Yes, DNA profiling. A child received DNA from both parents, and then they're saying that the profile of the mother, child, and the possible father are determined, meaning that we collect them. Ne? And then after that, the comparison of DNA bands of the mother and the child is made. The remaining bands, all the remaining bands, this is the key point. All the remaining bands are compared to that of the father. If all the remaining bands of the child matches with that of the possible father, then it will be a biological father. If all the remaining bands does not match, then it will not be the possible father. You understand? Yes. So that person will not be the biological father. So that is what you're supposed to explain. Life is good, man. Oh, this one, they brought it. They brought it. This, and I say, I don't care how do you read it. CADASIL, whatever. I'm saying the same thing. Whatever, I don't care. Is an autosomal genetic disorder caused by a dominant allele, and it is as a result of thickening of the walls of blood vessels. The diagram below shows. And we are bringing, we don't just bring, we bring. They are saying that the type of dominance, the type of diagram which is being shown there, this is what called a pedigree. How many offsprings for individual 1 and 4? Individual 1 and 4. Did I say 1 and 4? Did I read 3 and 4, man? 3 and 4 is 1. It's only one individual. That's individual 8. This question is the same question they asked. Individual is 1. The phenotype of individual 10. The phenotype. This person is normal. From the condition. No. This person, individual 10, is shaded, caused by a dominant allele. So if this person is suffering from CADASIL, whatever. I don't care. And that yes, it's called this by. So this person has CADASIL. And the genotype of 5. This one, you remember what we said. So if this is 5, and then this person is normal, and it's caused by dominant allele, automatically this person is gonna be small d, because the moment a capital D is there, the individual is gonna be affected with CADASIL. You understand? So it means that all these people, they are small d. All these people small d. Why? Because this condition is caused by a dominant allele. Therefore, I will say that the genotype of this individual is D, small d. We continue the next question. Using the evidence from the diagram, explain why 1 and 2 are both. Using the evidence from the diagram, explain why the individuals 1 and 2 are both heterozygous. Individuals 1 and 2. How many marks? 4 marks. Oh yeah, I see now. They want you to prove that why is this person. Remember, this person is affected by the CADASIL, ne? So, and it's caused by dominant allele. So this person can be capital D, or capital D, small d. That's what I did even yesterday. Capital D, capital D, small d. All right. So now, but they produce a kid who is small d, small d, small d, small d. So if this is small d, this can't be. If this is small d also, you see, this one also can't be. So it means that one came from the father, one came from the mother, and one came from the father. So automatically is individual D is small d, and individual this one, number 2, is small d. So they want you to put it in the writings. Then you're going to tell us individual 5 and 6 are normal from the condition. Therefore, they are recessive. They contain 2 recessive alleles. That is, you are saying that these individuals, they are homozygous recessive. Still is the same thing as saying 2 recessive alleles. Then you tell us that this, both of them, they obtain 1 recessive allele from either of the parent and the second recessive allele from another parent. You don't say that they obtain the recessive allele from the parent. They obtain 1 recessive allele from either from this person or from this person. And then the remaining recessive allele was obtained. If they got it from here, then the second one must be obtained from another parent. That's what they wanted. They wanted to show us that these two, they wanted the genotype of them, which is homozygous recessive, or they contain 2 recessive allele, and they wanted you to show us how did they become homozygous recessive. They inherited this recessive allele from the parents, meaning that they got 1 recessive allele from either of the parent and the second recessive allele from another parent. Hey, someone saying that this question was classwork. Yeah. Alright, Buffett. Hey, this one was your classwork. This one was our meal yesterday. If it was your classwork, it was our meal yesterday. It was our scope yesterday. Alright. Okay, we continue. What is the percentage chance of parents 7 and 8 having another child with CADASIL? What is the percentage chance of having another child with 7 and 8 to have another child of having CADASIL? So automatically this one is, since they have someone who is normal, is the capital D, small d, and then this one, since it's normal, is small d. So it's gonna be 50%, because if I say that this goes with that, is gonna be capital D, small d. This goes with that, is gonna be capital D, small d. This goes with this, is gonna be small d. Then this goes with this, is gonna be small d. So if it's 2 out of 4 for either this or this, and then times 100, so it's gonna be 50% chance. So the answer here is gonna become 50% chance. 50% chance. Someone said 100%. 100%, eh? How did it be 100% when this one is already sick? And then because they already produced someone who is small d, automatically this can't be like that. You understand? So if this was homozygous dominant, then automatically it would have been 100%. But it was not homozygous dominant, because of the individual 9 they produce, who is what? Who is normal. Yeah, is homozygous recessive. Okay, who got 9 out of 9? 9 out of 9. All right, we continue the next question. The next question is saying that, okay, blood group. Yes, we asked you a lot of questions on blood group, and they brought this in June, not June, in prelims of Gauteng. All right. The graph below shows the distribution of blood groups for 2 ethnic groups in certain countries during 2021. The percentage of people with each blood group. Which blood group? Which blood group is most common? Which blood group is most common in both ethnic groups? Most common. The percentage of with the blood group. So automatically it's going to be blood group blood group O, because if I add this and this, it's gonna be something. This one is a little bit lower, so the answer is gonna be blood group O. Has the highest percentage in ethnic group 1 than ethnic group 2. This one is C. 1 is lower than 2 is equal, lower than 2 is higher than 2, so it means that it's gonna be blood group A. It's gonna be blood group A. State the possible genotype of people in ethnic group 1. Guys, just a moment, a small moment here. I need to check up on the power. Okay, so we are saying that how is it not AB? They say the most common. How is it not AB? Which question is that? How is it not AB? The higher percentage, common. What did I say? Yeah. Yes. Let me see the question. What is saying? They're saying that is the most common in both ethnic groups. Oh yeah, man. Yes. They're saying that the most common, meaning that they have almost the same amount, which is 4 out of 4. So it's B. Thank you so much. Is A, B. Yes. Then they are saying that which blood group has the highest percentage in group 1 than in ethnic group 2. Group 1. Where is group 1? Yes, we say that. We said it is A. Yes, it's A. It's A. So I was rushing, ne? Yeah. If you said phylogenetic tree, no man, it's not phylogenetic tree. Even size confused. Yeah, no man. All right, we continue. Wait, now how can you say that? Okay. Akira, you are rushing me. They are saying that set all the possible genotypes of state all the possible genotypes of blood group of state all the possible genotypes of 11% of people in ethnic group. 11%. Where is 11%? Oh, 11%. This 11%, it shows that it is blood group B. But this blood group B can be homozygous or can be heterozygous. It can be homozygous or it can be heterozygous. So it means that I'm going to say blood group A, blood group B, heterozygous, or blood group B, homozygous, or blood group B, heterozygous. So that is the meaning. So we got the next question. So it means that they want you to write the genotype of blood group B, of which it can be blood group B. If you wrote it like this, 0, we're gonna give you 0 with the eyes. If you wrote it like this, 0. If you wrote it like this, 0. There is no such. So I told you that yesterday. Each group, ethnic group 1 had a population of 360,000 people in 2021. Which blood group A B or O? A B or O had this? Okay, now I don't know. I need to check. Okay, so I need to find out. So I'm gonna say 39,600, yes, divide by out of 360,000 times 100. Ish, guys. I don't have a calculator here. You know, I'm just doing the question paper just like you. Just like you. I don't know. Guys, I don't know. How much did you get there? I don't have a calculator here. I have to calculate. Let me check. Let me check and use my phone and see. Ish. Now I have to find out what is the answer, because if I don't... Okay, they're saying 39600 divide by 36000 0,11, yes, times 100, which is 11%, which is times 100, which is 11%. So I need to look at which one is 11%. Is blood group B. So the answer becomes blood group B. Blood group B. Wow, thank you so much, guys. You gave me 11%. Yeah, I didn't have the calculator. So, yeah, I also got 11% here. Thank you so much. Yes, it's 11%. All right. Then they're saying that, you know, guys, don't make me rush, because when you rush me, I end up reading the question wrongly and then give you wrong answers. And I can't do that. All right. Explain the important... Explain. By the way, if you see me that you have read something wrongly, you tell me on the live so that I'm able to check it before I continue. Explain in the inheritance of blood group AB. Explain the inheritance of blood group AB. Oh, okay. Now you need to understand. You need to understand what is blood group AB. What is blood group AB? Blood group AB is codominance. Wabon. Blood group AB is codominance. So is blood group AB. So you're gonna tell us that blood group AB is codominance, isn't it? Whereby it's A and B. It has both allele for A and allele for B. You understand? One allele, let me say, yes, one allele will come from the father, i.e. A or B, and the other allele will be inherited from the father, and the other allele or allele will be inherited from the mother, i.e. A or B. Why do I say A or B the same thing? Because I don't know which one is going to come from the father, which one is going to come from the mother. I don't know which is going to come from the mother. I don't know which one is going to come from the father. So one allele, yes, the person is codominant. Yes, they're going to hope to give you a mark there. And giving me the genotype of the person is AB, which is not the phenotype. The phenotype is given, but you want the genotype, which is AB. Then you have to show us where is this A coming from and where is this B coming from. It means that the A, one allele of either A or B will come from the father, and the remaining allele, either A or B, will come from the mother. That's how it's being inherited. Yes. Explain. So we continue the next question. Oh, they're asking you a question. Here. Name the type of dominance displayed in the inheritance of. Okay. Palomino. Hey, yeah, we did the horse. Yeah, Palomino are bred by horse breeder for their coat color, and then the Palomino has golden coat. Golden, okay. They're saying it is an offspring of the horse with the cream. It is an offspring. It is an offspring. Oh, I'm getting the question now. It is an offspring. This Palomino, whatever, I don't care. Palomino is an offspring of cream-colored coat. Okay, and the horse, which is AA, meaning that this one is AA. All right. And since they are both of them are capital letters, it means that it is co-dominance, or it is incomplete dominancy. Okay. They're saying that horse with chestnut colored coat. All right. It means that this one is gonna be small capital G, capital G. All right. Meaning that if this is a combination of the two from the offspring, it means it's going to be A and G. All right. Name the type of dominance which is being displayed. This is not a mixture. They say that Palomino is horse golden. Something golden. It's not a mixture of color. It's an intermediate of a color. Cream-like, and so since it is an intermediate, it is incomplete dominancy. That's why I gave you the differentiate between complete dominancy and incomplete dominancy on our scope. Yes. Explain the answer. So the answer, why do you say is incomplete dominance? It's because the Palomino, the offspring, yes, is heterozygous. Heterozygous. You understand? With an intermediate color, that is golden color, with an intermediate color. That's what you are supposed to look at. You need to show us that is heterozygous, and you also need to show us that it is incomplete. It is incomplete. Is incomplete dominancy. How? Heterozygous with an intermediate color. All right, meaning that it is a what? Is a middle color. All right, we are saying that breeding of horse with the desirable characteristics, okay, is a type of biotechnology. Breeding of horse with the desirable character, Oh, I told you that we only give. Okay, this question, it was breeding of horse. The question which I brought, it was about cows with milk. But I also brought the question of horse. All right, cow with milk. And then I say that breeding of horse is a type of biotechnology. Name the type of biotechnology which is being shown there. It is selective breeding. Yes, because the question is saying that explain selective breeding for the cow to produce more milk. Okay, selective breeding. So this one is going to be selective breeding. Then they are saying that if you say neither of the allele is dominant, neither of the allele is dominant. If you say neither of the allele is dominant, even the codominance is neither of the allele is dominant. They need to talk about intermediate in that question. In this question, you need to talk about the intermediate color and the heterozygous. Maybe that one they will correct you related to that, but also maybe they will give you 1 mark. Yes. All right, we continue to the next question. Name the 2 phenotypes of the horse that must be bred. That must be. Name the 2 phenotypes of horse that must be bred. Interbred for 100% palomino offspring. Remember, palomino offspring is a combination of the two. Then automatically I'm gonna cross this and this. Then automatically I'm gonna get the, if I cross A with A and G with G like this, automatically all of them is gonna be GA or AG, whatever. GA, GA, GA. You see, all of them 100% is gonna be palapino. And then you see that we say that palapino is AG or GA. It's the same thing. GA. You see, it's the same thing. You see, so it means that I have to cross. Your answer was supposed to be you have to cross cream colored coated horse, and then also with the also chestnut colored coat horse. We continue the next question. Use the net cross to show the ratio of offsprings. Show the ratio of offsprings. But they say the horse of cream color. Okay, they say that cream colored coat was crossed with palapino. Palapino is the second generation because you can't get it from the first generation. Maybe they will create accordingly. I don't know. But to me, I'll say P2, F2 because palapino is the second generation. Or even those who said P1, F1, they could create you also. So phenotype. I told you, you get it from the question. They say, what did they say? Cream colored coat horse crossed with palomino horse. Cool. Then we go to the next point. What do you say? You say genotype. Remember, palapino is GA, and then this one cream. We say that when you go back to cream is AA. Cream is AA. So now it must say meiosis must take place so that the gametes are separated. A, G, A. Then they say that this is called the it's right, but it's just not in the right position. This is the genotype. This is the meiosis, and then these are gametes, and then fertilization. Now fertilization must take place so that I form the offsprings. So in this case, you can draw a Punnett square, or you can do what I told you yesterday. Put a dot, put a dot, put a dot, put a dot, so that everything is in order. This goes with that. I'm gonna say GA, then this with that. It's gonna say AA, then this with that. It's gonna be GA, and then this with that is gonna be AA. So these are offsprings, F2 or F1 offsprings. And then now we come back here. What are they saying? They want show the phenotypic ratio. They want the phenotypic ratio only. So it's gonna be 2 GA, and then 2 AA. What is GA? GA is palapino. Palamino, whatever. And then the other one, 2 palamino, and then the other one is what is coated, what cream coated is cream. The other one 2 is cream. But what is the question? What is the question saying? What do they want? They said ratio. So it's gonna be 2 to 2, but you can't write it like this because this is not the simplest. So it's gonna be 1 to 1. So that is the perfect answer. 1 to 1. Who got it right? We got the next question. Yeah, I wrote AG is the same thing. It's the same thing. The order doesn't matter. Yes, the order doesn't matter because they didn't give you the order to use. Either AG or GA is still fine. If you wrote AG or GA, you wrote AG, is fine. It's fine. 100% fine. Even here, if you wrote AG, you wrote AG is still fine. 100% fine. All right. The question is done. Guys, at least who got 48? 48. 50 out of 50. Oh, I have someone who got 48. Ah, I see you got 48. Someone is saying now I have 100. 86 out of 100. Hey, we are shifting. But guys, the paper was fair, ne? 49 out of 50. Ducky. You guys, 98 out of 150 so far. Thank you so much, guys. So it means that you only lost 2. Wow. That's so good, ne? You are so impressing me. You are impressive. You are impressing me so much. Yeah, I think I must be there when they're giving awards for life science, ne? Yes, there in Midranda. Say yeah. Thank you so much, guys, for following us on YouTube channel and TikTok. Facebook is there, but they always hack it. So there are some people even asking money from people, yet it's not us. We will never ask you money. We will never ask you money. And if we ask you money, you buy something, you buy it online, or it must be the account for Thunder. If they are books, that's the only money. If you are requesting for the books, nothing else. We don't send money once WhatsApp. We don't. Please don't do that. All right. Theory of human evolution are based on similarities between human and African apes. We brought this question. Give 2 characteristics related to vision. Okay, you can talk about any binocular vision, eyes in front. Yes, they can talk about whatever. You can see color. They can see color. All that. Okay, they're saying that describe 2 differences between the jaw of a human ape. Jaw. They're saying jaw. So for humans, it's like this. C shape, U shape, C shape. And then African ape, rectangular shape. Palate, the palate. The jaw for human is less protruding. The jaw for African ape, more protruding. Actually, African apes are prognanthus, if you wrote prognanthus. Well, this one you can talk about is non-prognanthus. Done. The jaw, I think even the canines can be there. For the case of humans, small canines, they just look at the jaw. Basically, they are looking at the jaw, and then you give the answers based on what? On the jaw. All right. We got the next question. Explain the significance of position of foramen magnum, the shape of the spine, Okay, wait. The shape of the spine. Okay. Okay, they're saying that explain the significance of position of foramen magnum, the shape of the spine, and the pelvis. Guys, this is the question which I asked you yesterday. This one. Where is it here, by the way? Is here. This one, the factors which affect 3 factors. The way they asked you them, and then I showed you where to get them from the distinction material book. Distinction material book, it will never let you down. So these are the questions we're asking you there. Following, you see that all this you are talking about, they're asking you, and then I told you, take a screenshot. So you're saying that foramen magnum is in a more forward position to allow the spine connect or enter vertically. Pelvic girdle is short and wider to support the upper body weight. Spine is S-shaped to act as shock absorber to allow flexibility during movement. That's it. Nothing else. We got the next question. They're saying that. So, that is 6 marks. Mahala. This one, it was also there. How many genera are represented in the diagram? I said 1, 2, 3. 3 genera. 3. Give 2 examples of Australopithecus africanus species found in South Africa. Give 2. Give 2. Give 2. There is the question I brought. Give 2 Australopithecus. Give 2 Australopithecus africanus found in South Africa. And then, 2. I asked for 2, not 3. I know there are 3. I wanted 2. 2. All right. They're saying that Australopithecus africanus, there is Taum child, Littlefoot, or Mrs. Place. Okay, sharp. Give the average brain of species that was first used tools. The average. Remember, it's Homo habilis. That's what we said. This one, the average volume. So you're gonna say 609 milliliters. Calculate the percentage increase. This one I explained it even in paper 1. Say that I'm not going to talk about paper 2 because you know. It said that final minus initial over initial times 100. Okay, final. Where is final? For Homo habilis and the Homo sapiens. Homo habilis. Where is Homo habilis? Homo habilis 609. 609. 6. Aibo. Final. Where is our final? Final is 1,333. 1,330 minus 609 over 609 because initial times 100 times 100. So what is the answer? I don't have a calculator, I told you. Why am I the only one seeing your scope after the paper? But I nailed it. It's because maybe you don't keep yourself on ThunderEDUC. You must keep yourself on ThunderEDUC with these scopes from grade 8. Yeah, grade 8. Yeah, so basically, sorry if you just saw it, but I know that at least you have been on Thunder. They're saying that I don't have a calculator. You guys, who got it? I will never skip your video. Okay, guys, let me calculate. I don't have a calculator, guys. If you have a calculator, okay. How much did you get, you guys? How much did you get? You say get 180. 118.39. Oh, so I see everyone is getting that. Yes, remember last time when I telling you that the percentage increase, it can be more than 100%. Yes, let me see what is the double of this. If I say this is the double of this, yeah, obviously is 1,218. 18. This is more than this, so automatically it doubled and then went up. So it's supposed to be more than 100%. So from the chart they're saying it is 118.39. Since everyone is getting that, then automatically it could be the answer. So if you calculate with the calculator, guys, I don't have a calculator. I'm also doing the question the way you are doing it. When they open, then you start doing. All right. Thank you so much, guys. Let's go to the next question. Is it the last question? Still have too many questions. All right. They are saying that the extract and diagram below are based on the ratios, whatever, breeding month of ratitis, whatever. I don't know. Okay, these are laying flightless birds. The eggs laying month are influenced by the month factor such as temperature, length of the day, availability of food. Ostrich normally lay their eggs in September and whatever between October and March, while imps lay their eggs. I remember when they brought this question some time back. Yeah, it killed the people. I thought, I hope it's not gonna kill you, this one. Then they're saying that state what is meant by biological species. Remember we said what's a species is a group of organisms that have similar characteristics that are able to interbreed naturally. Naturally to produce, they're able to interbreed naturally because of the biological to produce fertile offspring. Yes. Then they're saying that use the information in the extract explain why ostrich and emus considered to be different species. So they consider to be different species because they are supposed to be, okay, they are supposed to be able to interbreed. So in this case, one breeds in September, another one breeds in November. Automatically they can't breed. So they cannot breed. So they are different species. So they cannot interbreed to produce a fertile offspring because they are breeding at different time of the year. Yes. So that could be, yes, that could be the answer, that basically they lay their eggs normally in different times of the year. One is in September and then another one is in November. Lays its eggs in November. We got the next question. They're saying that you skipped a question. Sir, you skipped a question. Oh, guys, tell me which question did I skip? So this one is done. And also this one is done. Hey, guys, tell me which question did I skip? Oh, thank you so much, guys. I've seen it. I've seen it. Okay. Should I first finish this? I go back, or I should first... Okay, let me go back. Explain the relationship of average brain volume and the cultural evidence in human evolution. Remember, as the brain volume increases, there will be in advancements in the what? In the cultural evidence. You understand? Yes, I brought this question yesterday from our scope. Say that and they also brought it in this year in May, June. Say that explain the relationship of the average brain volume. As the average brain volume of an individual increases, yes, it increases the intelligence. You don't just link it directly because the brain volume doesn't mean anything to the technology you're doing. As the brain volume increases, the intelligence also increases. So it gives the individual more thought, hence an advancement in the tools which are being used. Yes, so you were supposed to explain it that side, that as the brain volume increases, still I skipped another one. Basically, I skipped the whole question. As the brain volume increases, okay, I'm gonna go back to it. As the brain volume increases, all right, there is more brain cells, and then this will increase the intelligence of an individual. Yes, hence advancements in the tools which are being used by human. Which shows that the advancements in the tools will show that there is an increase in the brain, in the intelligence of an individual. You understand? So it is the brain which determines the intelligence and which determines the tools, not the tools determining the brain. Nope. Yes. The brain decomposes after it dies. Okay. Explain why scientists are able to determine the brain volume of the fossils. Explain. Explain how the scientists are able to determine the brain volume. Oh, yes, I brought this question. They're asking you, but I brought it. How is the cranium related to? So the more, it means that when it decomposes, it leaves the skull, it leaves the cranium now, because we're talking about the brain. It leaves the cranium. So the scientists explain how scientists are able to determine the volume of the brain. It means that they have to determine the space in the cranium. So as the cranium increases, yes, it means that the number of brain cells to be accommodated also increases. You understand? So let me show you what here is the answer. Here is the answer. Ne? But now check that if the cranium, now this one is out, it's not there, and also this one is out, the brain is not there. You understand? So but you see that the skull, the cranium, not the skull, the cranium, the space of the cranium, the volume of the cranium will determine how much brain the individual is having, because we cannot trace the brain of 2 million years ago individual. No, but we'll trace the skull because the bones will be still there. So the scientists use the volume of the skull, and then to determine the volume, to determine the size of the brain. You understand? Therefore, now they can link it to how big the brain was. I think now, yes, I hope I have not skipped any other question. Give to average was first used tools. This I answered that. I also answered that. Okay, thank you. We continue the next question, guys. I stopped here. State 2 other factor of reproductive isolation that would have prevented this and this from interbreeding if they were the same habitat. Oh, if they were the same habitat. First of all, breeding at different time of their different seasons. Obviously, even if they were the same habitat, they wouldn't have breed in the same time of their. And then we can also talk about they could have been maybe, did I say from the extract? Nope, they didn't say from extract. Different courtship behavior. Different courtship behavior. The infertile offspring. Yes, this. Prevention of fertilization. Different courtship behavior. Breeding at different times of their. Did I say other than? Let me say. Give to other reproductive isolation mechanism. Okay, so you don't talk about different season, because here they talked about different season, so you don't talk about that. So it means that you're gonna just talk about different courtship behavior and then prevention of fertilization or resulting in or infertile offsprings. We cannot talk about pollinating agent. Are they flies? They're not flies, so you don't talk about different pollinating agent. Yes, so you only talk about this, you talk about this and this. That's it. Then we are saying that identify the evidence from evolution represented in the diagram above. Identify the evidence for evolution represented in the diagram above. Oh, evidence. Let me read that again. This is a geographical isolation mechanism, distribution of organisms in different parts of the world. Geographical isolation. Yes. This is geographical isolation. That is the one evidence of evolution. Yes. Distribution of organisms to different parts of the world. Okay. You're saying that what if you said geographically? What if you said geographically? I don't understand what you're saying there, but maybe they will give you. But it's a geographical isolation. Oh, geographical, they are being separate geographical. I don't know. Okay. They're saying that identify the evidence. Okay, we're done. Describe how the geographical isolation. How is this geographic isolation support the theory of evolution? So automatically, now here you have to explain to us how did they distribute themselves, and then to become these different organisms. Yes. It's there. It's there in our hey, it's there. I wrote it here. Where is it? Yes, I wrote it there. Yes. So starvation resistance refers to the period for which organisms can go without food before it dies. An investigation was conducted to determine the effect of food the availability of food on the starvation resistance in the fruit flies. They're saying that the procedure was as follow: 500 fruit flies of the same age. Okay, let me just read just fast so that I'm able to understand what is here. They were all placed in the container of growth. Were given in cellulose. You can read yourself, ne? Me, I'm reading to understand. The food source was removed after an hour. Okay, they took 8% of the fruits. Okay, to die from starvation, and recorded when did you record? Okay, the flies were removed from the container. Okay, so that they clear up. Food sources were returned. The remaining, so they repeated. Okay, you have transferred to the new container. The first fruit flies were allowed to grow in the same age of the first generation. The food source were the time taken for the fruit fly to die. Starvation was recorded. Yeah, someone is okay. Let me first finish up by procedure represent. Yeah, someone is asking me if I wrote by geography here. Identify the evidence shown in. Yes, by geography is the same thing. Geographical isolation or by geography is the same thing. They will still give you. Yeah, by geography still is fine. All right. State the independent variable in this scenario. Okay, I'm seeing it. State the independent variable for this scenario. So in this scenario, the independent variable, yeah, they said it. They talked about the aim here. An investigation was conducted to determine the effect of availability of food on the starvation resistance in the fruits, meaning the availability of food will be the independent variable. Then they are saying that describe how the dependent variable was measured. How did they measure the dependent variable? How did they measure the dependent variable? They said it here. The time taken, the time it took for 80% of the fruit flies to die from starvation was recorded. You see? Time taken. That's how they measured it. We got the next question. State one control variable that was considered when selecting the fruit flies. What variable did they talk? They talked about. They talked about the age, same age. They used the same age, so it was this the control variable which they used. We got the next one. Harvard, here I come. They're saying that explain why the investigator gave the same food. Why did they give them throughout? Basically to minimize the interference from the other food, because the other food could be having different content and might result in different effects. Yes, so basically they wanted to bring about the validity of the results. Yes, these are questions where the answer might not be one. You understand? The answer might not be one. Then they got the next question. It's almost done. Then they are saying that the graph below shows show the results obtained in the first generation and then 60th generation. Okay, describe the result obtained in the investigation. Oh, they're saying describe. Just tell us what is happening. So basically, you see that in the first generation, we see that there are many died, many flies died between 0 and 40 what? 40 hours. Between 0 and 40 hours. In the second, in the 60th generation, many flies died. Yes, number of, let me check here, hours until death from starvation. Meaning that here, all this time they didn't die. Here they didn't die, they only started to die here. Here, all this time they didn't die. So here it means that many died between maybe 140 to 180. Yes, after between 140 to 180 hours of starvation. Here, many died between 0. Is it between 0? Maybe between 20 and 40. Does it touches to the no, it's not 0. Between maybe 20, between 10 and 40. Yes, many of them died after those hours of starvation. But here, you just have to describe your English. Then they're saying that use the Darwin theory, explain the increase in the starvation. Okay, here they want you to use natural selection, the principle of natural selection. Still, is the same story here, which I gave you here. Yes, here the principle of natural selection. So the other one was speciation. Now this one is natural selection. So they brought both of them. I know that you guys, you got it, so I'm gonna try to explain it. So those who explain by geography here, how did those new species form, you will get your marks. Yes, 5 marks using speciation. And then here, those who used their evolution by natural selection, they're also going to get their marks. But here, you have to tell us that you have to apply. Even there you have to apply. Using, basically flies produce a large number of offsprings. There is variation among these flies. You understand? Those which are resistant to starvation. Yes, they said they're resistant to starvation. Yes, they said they're resistant to starvation. You see, those which are resistant to starvation and those which are non-resistant to starvation. In the first generation, after exposed to starvation, yes, those which are non-resistant to starvation, they died in large numbers. While those which were resistant to starvation, they survived. Those which are resistant to starvation, they pass the allele of resistance to starvation to the next generation. You understand? After many generation, there was a high proportion of flies which are resistant to starvation. You understand? Than those which are not resistant to starvation. So basically, that is it. I think, guys, that is it. I hope you guys, you got all marks. Thank you so much, guys. Let me see people from the comment section. What are they saying? Is speciation in the speciation? I forgot to include no gene flow. Yes, you lose a mark. Then there's someone geographic isolation and speciation are the same. Yes, it is the same. Basically, speciation is divided into two. We have speciation by geographic isolation and speciation by sympatric, which is it could be distance or it could be seasons, distance, like that. So in this case, we only describe geographical isolation, which results in by geography. the organisms deciding to differ. Oh, Mr. Saidi, will you always be famous, my dear? Thank you so much, guys. Thank you so much. Thank you so much. Who is this one? Specific Buteles. Specific Buteles, you have been with me for so long. For so long. Specific, where do you stay? I think I should send you something from Thunder. Where do you stay? Or try to access my numbers for Thunder. Any number from Thunder, then you ask for Mr. Saidi. Then maybe we will send you something, because I've seen you for so many, since January you are with us. Thank you so much, guys. You guys, you rock. You guys, you rock. I see the Max Day happiness I'm having here. I got a distinction because of you. Lolo. Thank you so much, Priscilla. 1-4 out of 150. Guys, I wish you get all of you distinctions. You remember it was 100% distinction. We want distinction project, 100% distinction. Thank you so much, guys. I got 90%. That's good. It has been 2 years, sir. I'm from Gauteng. Tetilo Secondary School. I think, I don't know. Let me just take a screenshot of it. I will try to follow it up, but try to check upon me so that I'm able to maybe send you something from Thunder. Anything which is written as a proud subscriber from Thunder. Yes. See you in another life, guys. See you too, guys. This is my prayer for you. I pray that you guys, all of you, you pass. I pray that when the markers are marking, they see correct answers only. I pray that you get a lot of money, which is blessed money. I pray that you enjoy your life. I pray that you get a healthy life, you and your family. And I pray that you become happy when the marks come back. See you until you see me again. See you when I'm driving as you are driving. Amen, 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 amen. Guys, I think I'm done for today. I'm done for the year. Yeah. So it means that from now, I need to prepare for my holiday trip. So that yeah. All right, guys, it has been a wonderful year. Our prediction or our scope was nice. For paper 1 it was nice. For paper 2 it was nice. If you have sisters and brothers, you can also refer to them so that they also be able to benefit. So guys, we are going back to geography. Thank you so much, guys.